Hello friends, welcome back to my YouTube channel, The Microbiology Digest. Today I'm back with a new topic of immunology, which is very important to understand. That is the antibody, its structure and functions. So as we all know, antibody and antigen interactions are the basis of the science of immunology. So in response to an antigen, our body mounts two types of immune response. One type is called as humoral immune response, which is mediated by immunoglobulins or antibodies, and second type is called as the cell mediated immune response. So now the antibodies, our topic of discussion today is about the antibodies. So antibodies are also called as immunoglobulins. They are glycoproteins produced by plasma cells, and these antibodies or glycoproteins are produced in response to an antigen. These are produced by antigen sensitive B lymphocytes when they encounters a specific antigen, and they have the property, these glycoproteins, they have the property of binding specifically to the antigen against which these antibodies or glycoproteins are produced. These molecules, antibody molecules, they plays a very important role in the humoral immunity, and they recognize specifically the antigen molecules or the microorganisms, and they can bind specifically with them and thereby helps to eliminate or destroy these microorganisms from our body. They are also integral part of the adaptive immune system, because antibodies, they are part of adaptive immune response also, and they helps in elimination of the foreign molecules by specific recognition, or otherwise also true by specifically sensitized T cells. Now, as I said, antibodies are also called as immunoglobulins. Why? These two terms, antibodies or immunoglobulins, these two terms are used in most cases interchangeably. So what happened? Two scientists, Tiselius and Kabat, they subjected the serum to electrophoresis, and they could recognize four different fragments. They named it as albumin and globulin. Three fragments of globulin were recognized: globulin alpha, beta, and gamma. And these antibodies or glycoproteins, they were found to be located in the gamma globulin fraction of the serum, and because of this they are also named as immunoglobulins by J.F. Harrimans. So immunoglobulins, they account approximately 20 to 25% of the total serum proteins. So out of four fragments, the immunoglobulins, they approximately they cover around 20 to 25% of the total serum proteins. So antibodies or immunoglobulins, they are almost same. Both the terms are used to understand the physiological as well as the biological properties of the antibody molecules. Now let us understand the structure of an antibody molecule. So the structure of the antibody molecules, it was elucidated by Edelman and Porter, two scientists. And because of this great discovery, they were awarded Nobel Prize in the year 1972. So an antibody molecule, it is a typically a Y-shaped structure, Y-shaped molecule. It is a tetramer composed of four polypeptide chains. So these four polypeptide chains, out of these four polypeptide chains, two identical chains, short chains, they are called as light chains, and two identical long chains, they are called as heavy chains. So light chains, it is approximately 220 amino acid long, and they are attached to the heavy chain by means of disulfide bonds. The heavy chain, in contrast, it is a bit long, as I said, it is 440 amino acid long, and it contains variable domain as well as the constant domain. The light chain, there are two types of light chains are available. They are kappa and lambda chains. And five different types of heavy chains are there. They are gamma, alpha, mu, delta, and epsilon. So each heavy chain class, they represents the antibody isotypes. So molecular weight of an antibody molecule, it is around 180 to 900 kilodaltons. So this is a typical diagram of an antibody molecule. You can see it is an Y-shaped molecule having two light chains. The short chains are called as light chains, and the long chains, they are called as heavy chains. It has two terminals. One terminal is called as N terminal, and other terminal is called as carboxyl terminal. So we'll be discussing, it has different components. We'll be discussing one by one. First, let us discuss about the heavy chains. So as we have already discussed, there are five different types of heavy chains. Heavy chain classes are there. They are named as gamma, alpha, mu, delta, and epsilon. So each heavy chain class, it signifies the antibody class or antibody isotypes. So gamma, antibody heavy chain gamma, it represents the immunoglobulin G. Alpha, it denotes the immunoglobulin A. IgM or immunoglobulin M, it is having the mu type of heavy chain. Then IgD, it is having the delta type of heavy chain, and IgE having the epsilon type of heavy chain. Now coming to the light chain. Light chain, it is of two types, kappa and lambda. Each antibody molecule can have either two kappa or two lambda chains, but they cannot occur both at a time. And the proportion of kappa and lambda chain, it varies according to different species. So in case of bovines, the ratio of kappa and lambda chain, it is 10 is to 90. Then in rabbits and mice, it is 90 is to 10, just reverse. In case of swine, it is 50-50. Equine, it is 5 and 95. In case of human, it is 70 is to 30. So this is again another structure of antibody molecule showing different domains. So these functional discrete sites, these are called as domains. Okay. So these are light chains, they have two domains. So whereas heavy chains, they may have 4 to 5 different domains, and this region is called as hinge region. So we'll be discussing the specificities of this region in our subsequent parts. Now, as we have discussed different domains, two types of domains are available: constant domain and variable domain. Amino terminal or amino acid sequences of two distinct regions, both in heavy as well as light chain, they are named as variable as well as constant. The N-terminal region approximately 110 amino acid long, and the C-terminal, it is a bit longer, it is 330 amino acid long. Then in heavy chain also, in light chain also, just like heavy chain, they are also having uh, two different regions. One region is called as constant region. It is written as constant L, means constant on light chain, and the variable regions, which is written as VL. And these regions, this is a polypeptide loop of around 110 amino acids. These variable domains of both the chains, both heavy chain and the light chain, they fall together to form a single antigen binding site. So antigen binding site is located in the N terminal of the antibody molecule, and this is formed by the variable domains of both light chain as well as the heavy chain. So as the antibody molecule, it is having a Y-shaped structure. So each antibody molecule will have two identical antigen binding sites. Okay. The other part, they are called as, they are conserved and they are constant domains, and they are having different biological activities. So we'll be discussing later. Now another term that is called as CDR. This is a portion. CDR means it denotes the complementarity determining region. It is a part of the variable region of both heavy chain as well as light chain. So CDR is usually, it is formed by the variable region of both heavy chain as well as the light chain. It is a very short stretch of amino acid, usually 6 to 10 amino acid long, within the variable domain, and these sequences are highly variable, and these regions, they are also called as hypervariable region or complementarity determining region. Why it is so? Because they has these sites are the sites which will exactly recognize the antigen molecule, or antigen will bind specifically to this region. Next is hinge region. So hinge region, this is a region which is present in gamma, alpha, and the delta heavy chains, and this region is present between the Fc as well as Fab regions of the antibody molecule, and this is generally targeted by the pathogen. This region, it is highly rich in proline and cysteine amino acid, and cysteine residues are mostly involved in the formation of interchain disulfide bond, whereas proline, it is responsible for prevention of the folding of the globular structure. So it is present between the CH1 and CH2 domain and confers flexibility to the antibody molecule. Because of this hinge region, antibody molecules are, they attains the flexibility. Okay. Next two regions very important, that is Fc region and Fab region. So these Fc and Fab regions, the structure of the immunoglobulin molecule when studied by digestion with various proteolytic enzymes. So two regions could be identified when the anti-immunoglobulin molecule was digested or treated with papain. Two identical portions are called as FAB, that is called as fraction antigen binding or factor antigen binding, and other portion was named as FC, that is fraction crystallizable. So when we digest an antibody molecule or immunoglobulin molecule with papain, so it will yield two identical FAB and one FC portion. Whereas pepsin, another proteolytic enzyme, it will yield two major fragments. One is two FAB portion will be joined together and one FC fragment. Okay. So FAB region is responsible for the antigen binding, and it consists of the entire light chain, both variable as well as constant, and the variable heavy chain, and the first domain, constant domain, CH1 of the heavy chain. The variable regions, they are mostly responsible for the specificity of the antibody molecule towards an antigen. Then Fc portion is responsible for mediating all the effector functions. So antibody molecules, they have different effector functions. So Through their surface receptors, various effector functions will be mediated by Fc portion of the antibody molecule. Now, this is diagrammatic illustration. In the first, you can see when we digest with papain, so two identical portions of these two are identical FAB portion, and this is the Fc portion. But same in immunoglobulin molecule, when we digest with pepsin, it will yield only two components. Both FAB portions are joined and one Fc portion. Okay. Now coming to the antibody isotypes. So as we have discussed, there are five different classes of heavy chains, and based on which the antibody isotypes are determined. So there are five major classes of immunoglobulins: immunoglobulin Z, immunoglobulin M, immunoglobulin A, immunoglobulin D, and immunoglobulin E. So again, each antibody isotype or immunoglobulin isotype, they may have further subclasses. Suppose IgZ, in case of cattle and buffalo, it has two subclass, IgG1 and IgG2, and in other species there may be more number of subclasses. And in case of chicken, IgG, it is written as IgY, and this is actually the counterpart of IgG in case of mammals. In case of chicken, IgY, it is the counterpart of IgG in other mammals. Now first let us discuss the immunoglobulin G. So it is the major antibody. Immunoglobulin G is the major antibody, and it is produced in the secondary immune response. Okay, and it is having a relatively small size, and it can cross placenta. Okay, and it is the most abundant antibody in case of in blood. It is found in the highest concentration in blood and other fluids like lymph, space, cerebrospinal fluid, and peritoneal fluid. It occurs in monomers or monomeric form. It can opsonize, agglutinate, and precipitate antigen. It has multiple functions. It can also activate the complement cascade. What is complement? We will discuss in our subsequent discussions. And there may be different subclasses of, as we have already discussed, the IgG molecule. It may have different subclasses depending on the species. For example, in cattle and buffaloes, it is having two subclasses, IgG1 and IgG2. Next is IgM. IgM, it is a major immunoglobulin produced in case of primary immune response. Okay, it is the major predominant antibody molecule produced in case of primary immune response, and it can provide the protection against the invading antigen till sufficient quantities of IgG is produced, because IgG is produced in case of secondary immune response. So it is having a large size molecule, and it is mostly confined to the blood vessels and accounts for approximately 10% of the total immunoglobulin pool of the body. It is present in the second highest concentration, and it occurs as a pentamer. It is having five different chains. It occurs as a pentamer, and it is having an additional structure called as J chain or joining chain. So J chain is present in IgM. There is no hinge region. Hinge is absent in case of IgM. IgM monomers are linked together by disulfide bonds and J chain in a circular fashion. So in the diagram, we will see how IgM molecules are structured. Our monomers of IgM are joined together in a circular fashion by J chain. Okay, it can activate complement fixation. It is It can also exert its effects by mediating phagocytosis, then complement activation. It is also having a lot of biological functions. Next is IgA. IgA, it is a predominant immunoglobulin present in case of mucus secretions or body fluids such as milk, colostrum, saliva, tears, sweat, all secretions and excretions of the body. It will have IgA, and approximately 80% of IgA it occurs as a monomer, and it also occurs in a dimeric form. It is having two chains again as well as joined together by J chain, just like IgM. There's a J chain, and the it is also having different effector properties. It can activate complement. It can fix complement. It can exert its effects on the local invading organisms in the body surface, and the secretory component which is present, it mostly protects the mucous membranes of our body. Next is IgE. This is also known as reagenic antibody because it is present and it is produced in response to allergies. It mediates the hypersensitivity reaction, and it is present in very low concentration in the serum, and mostly it is found on the basophils and mast cells and plays a major role in the immunity to parasites. Next is IgD. It is present in large quantities along with IgM, and it accounts for less than 1% of the total serum immunoglobulin. It occurs as a monomer, and it is having a very high carbohydrate content, which is due to presence of N-acetyl glucosamine. Okay, it has only two domains, IgD, and heavy chains of IgD. It is having only two domains, and it lacks the CH2 and has a very long exposed hinge region. Hinge region in case of immunoglobulin D, it is a very long region and it is exposed. Okay, and this is very much labile immunoglobulin. It is easily degraded by the heat and the protein. It can be degraded by proteolytic enzymes. So these are some diagrammatic illustration of structures of various immunoglobulins. So this is IgG, this is IgD and IgE, and this is IgA. It is a dimer, and this is the secretory component, and this is the J chain, and this is the IgM which exists as a pentamer, and this is the J chain. Okay. Now, physicochemical properties of different classes of immunoglobulin molecules are very important. So first property is the prototype, as we have already discussed. Z is the monomer, IgA is dimer, IgM is pentamer, D is monomer, and E is again a monomer. So valency, depending on the type, the valency or the ability to bind antigen, actually it varies. IgG, it will have two antigen binding sites. IgA, it will have four. IgM, as it is a pentamer, it will have 10 different antigen binding sites. D and E will have two antigen binding sites each. So likewise, different properties of each antibody molecule have been listed out here, and these are very essential to understand, and if possible you can remember. So here IgG, it can already we have discussed, it can cross placenta, but here one exception is there. IgG2 subclass cannot cross placenta. Okay, so out of all the subclasses, IgG2 subclass of immunoglobulins, they cannot cross placenta. Okay, Now coming to the antigenic determinants. So based on the antigenic determinants present on the immunoglobulin, there may be of three different categories. So isotypic determinants, allotypic determinants, and the idiotypic determinants. So isotypic determinants we have already discussed. They vary from each other in the amino acid sequence of the constant region in the heavy chain. So these variations are called as isotypic variations, and based on these variations, we have five different classes of antibody molecule. Each antibody class they are called as isotypes. Next type is the allotype. These are determinants in the immunoglobulin molecule that differ among individuals of the same species that have inherited different alleles, and they are called as allotypes. So antigenic determinants present in different isotype genes in the constant region of heavy and light chains, they are encoded generally by multiple alleles, and these changes they may lead to form different antigenic determinants, and these allelic changes they are collectively termed as allotypes. Next is the idiotype. Idiotypes are again these are due to the mutation in the genes of the variable region, mostly the somatic hypermutation. Idiotype may act as a foreign to the host sometimes. So there may be little or minor somatic mutations, and each idiotype they behave like a foreign. If there is some mutation, it may behave like foreign to the host. Now coming to the biological properties of immunoglobulin molecule. So immunoglobulin molecule they serves lot of functions. Biological uh, functions are there. Primary function is the antigen binding, which is mediated by the FAB portion of the antibody molecule. And secondary functions are the complement components, fixation, then activation of various cells of the immune system. So important biological functions are protection, placental transfer, and the cytopilic functions. IgG confers immunity to most blood-borne infections. IgM mainly confers protection in the blood and plasma. IgA is the mucosal surfaces. Already we have discussed these properties. And similarly, IgE is the antibody mostly associated with the allergic conditions. So likewise, each and every immune antibody class, they are having different biological activities. So these are points I wanted to discuss today about the antibodies. If you find these informations are helpful, please subscribe to my channel. So with this, thank you all.